Here we could see a picture which depicts the diakinesis. Diakinesis state. That means V-shaped structures are there. Then this is a picture of meiosis. So the second one, double crossover is happening. Then leptotin stages. Then diplotin. Zygodine stages. Pachytene, X-shaped structures are there. And prophase 1 stage, leptotin, zygotine, pachytene, diplotin, and diakinesis. Okay. Grasshopper test is squash preparation to study various meiotic stages. Aim to study different stages of meiotic division during spermatogenesis of grasshopper. Materials required: male grasshopper, needle, compound microscope, spirit lamp, acetocarmine, insect saline solution, HCl 0.1 to normal. Canoise fluid. Procedure: collect male grasshoppers, place the abdomen in insect saline solution and slowly cut the posterior segment and press gently. The testes will come out as a bunch. Then the testis lobes are transferred into canoise fluid and keep it for 1 day. After a day, the testis lobes are placed on clean glass slide. It is then hydrolyzed using 0.1 normal HCl and heated gently. Then it is washed with water. Apply 1 to 2 drops of acetocarmine stain with a sharp blade, and testis lobes are cut into minute pieces and kept for 20 minutes. The slide is then gently covered with a cover slip, taking care so that air bubbles are not formed. Then warm the slide gently and place it between 2 folds of filter paper. Press the material with the tip of finger and remove the excess stain which comes out on the sides of cover slip. Then the slide is observed under the microscope. Observation: different stages of meiotic division were clearly observed. First, interphase. This phase is usually present in all animal cells. The cells in this stage are physiologically active. No DNA replication takes place. No DNA replication in interphase. This phase is usually present in all animal cells. The cell in this stage are physiologically active. No DNA replication takes place. The cell undergoes active genetic and metabolic preparations. No DNA replication. The period of this preparation is called interphase or preparatory phase. It has 3 substages, G1, S, and G2. Prophase 1. The first meiotic phase is most prolonged, elaborate, and highly complex. It consists of 5 substages. First, leptotin. The chromosomes are long, slender, and uncoiled. They have a bead-like appearance. The chromosomes are long, stranded, and uncoiled. They have a bead-like appearance. They are densely formed on sides of the cell. Only one sex chromosome occurs in the male, which normally replicate later and hence appears as a dark skin body. Then zygote. Homologous chromosomes pair by a process called synapsis. Pairing starts from many parts of the chromosome. Pairing starts from many points on the chromosome. The chromosome are called bivalents. Bivalents become shortened and thickened by coiling and condensation, and synapsis of chromosome is converted by a complex called synaptonemal complex, which facilitates crossing over in case of zygote. Then pachytene crossing over takes place between non-sister chromatids. Crossing over is accompanied by the chiasmata formation. Chromosomes get shortened and thickened due to condensation of chromatin. Then diplotin. The homologous chromosomes start to separate and condensation progress successively to a greater extent. Each chromosome can be distinguished separately. Then diakinesis, homologous chromosomes begin to coil and become shorter and thicker. Chromosomes are fully contracted and deeply stained. The X chromosome is rod-shaped, univalent and easily distinguishable from the net chromosomes. Metaphase 1. The chromosomes are at the equatorial section of the spindle, and then centromeres are attached to the chromosome fibers. Each chromosome is easily seen. Maximum concentration occurs at this stage. Anaphase 1, the spindle fibers contract and the homologous chromosomes separate and move towards the opposite poles. Each chromosome consists of two chromatids attached to one centromere. Then telophase 1, the separation of homologous chromosomes is completed. Any sets of opposite poles, two distinct daughter nuclei are formed. The daughter nuclei formed contain only half the number of chromosomes present in parallel cell. Cytokinesis may occur after some time, after the completion of telophase. Meiosis 2. The second meiotic division is very much similar to meiosis prophase 1. The chromosome with the chromatid becomes short and thick. The nuclear membrane and nucleolus are absent. The spindle is formed and chromosomes are arranged in equator. Metaphase 2. The chromosome undergo condensation, moving to the center of the cell, attaching to the spindle fiber and form an equatorial plate. Anaphase 2. The spindle is formed. The centromeres of the daughter chromosomes are attached to spindle fibers, and groups of daughter chromosomes in each cell have started moving apart towards the opposite poles of the spindle. In telophase 2, two groups of daughter chromosomes in each haploid cell have surged the two poles of the spindle. The two haploid daughter cells formed as a result of meiotic division divided again by the second meiotic division. Four haploid cells are formed from a single diploid parent cell. Significance of meiosis: forms an essential prerequisite for normal sexual reproduction in plants and animals, brings about redistribution and reorganization of the genetic material through crossing over and recombination, causing the shuffling of genes and the formation of new gene combination. This in turn creates genetic variability, promotes the adaptability and fitness and survival value of organisms, and increase the evolutionary potentiality. Provide a mechanism for precisely regulating the chromosome number to a haploid value in gametes. Enable the combination between maternal and paternal chromosomes and genes in gametes. Stages of meiosis: interphase, plasma membrane, centrioles, cytoplasm, nuclear envelope, nucleolus, chromatin network. Prophase 1, leptotin, centrioles, nuclear envelope, homologous chromosomes, and nucleolus. Zygotine, asters, cross pairing of homologous chromosomes. Pachytene, diplotene, diakinesis. While drawing this, we need to remember something, that is, length should be reduced as stage advances and thickness should be increased. So thickness, in case of thickness, the most thickest one is going to be diakinesis. The reverse is the case with length. As the stages progresses, first stage will have the minimum thickness but maximum length. Next experiment: determination of respiratory quotient. To determine respiratory quotient, to estimate O2 consumed and CO2 released. The freshwater fish consumes O2 dissolved in the freshwater for cellular respiration and to release CO2 as a waste product by metabolic activity. The relation between O2 concentration of consumed and CO2 released is an intensive pattern of activation in the cells, and it is expressed as respiratory quotient, RQ. RQ is equal to volume of CO2 by volume of O2 into weight fish. Once again, RQ, respiratory quotient of this is equal to volume of CO2 by volume of O2 into weight fish. Volume of CO2 by volume of O2 into weight fish. Okay. And RQ around 1 implies that cell mainly catabolishes glucose. 1 in the sense it's glucose. Its RQ value around 0.71 implies the cell mainly catabolizes fatty acids. Its RQ value around 0.71 implies cell mainly catabolizes fatty acids, and RQ value around 0.3 implies the catabolism of amino acids. And RQ around 1 implies that cell mainly catabolizes glucose. If RQ value around 0.71 implies the cell mainly catabolizes fatty acid, and RQ value around 0.3 implies the catabolism of amino acid. 0.3 amino acid, 1 glucose, 0.71 lipids or fatty acids. Once again, 1. How to calculate RQ? RQ is equal to volume of CO2 by volume of O2 into weight of fish. Okay. Then RQ around 1 implies the cell mainly catabolishes glucose. If RQ value around 0.71 fatty acid, if RQ value around 0.3 implies the catabolism of amino acids. Materials required: fishes, jars, glasswares, Na2S2O3, sodium thiosulfate, MnSO4, alkaline Ki, Na2CO3, 0.025 normal. Once again, I am going to read. Fishes, jars, glasswares, we need Na2S2O3, sodium thiosulfate, MnSO4, alkaline Ki, Na2CO3, 0.025 normal, concentrated H2SO4, phenolphthalein. First, collect water sample from a pond. Estimate the amount of O2 and CO2 in it. Care should be taken. Do not agitate the water or to keep the level of CO2 content almost constant. Fill the rectangular jar of suitable size completely with sample water. Transfer a healthy fish of appropriate size, 2 to 3 inches. Closed the jar and covered the whole setting with black paper. Keep it for an hour. After this period, remove the fish. Estimate O2 and CO2 present in water. The difference in the initial and final reading gives the amount of O2 used and CO2 released at room temperature. Calculate reciprocity quotient using this formula. N1E1 is equal to N2E2, where N2 is equal to N1 into V1 into equivalent weight of O2 into 1000 by V2. Once again, how to calculate this? N1V1 is equal to N2V2. N2 is equal to N1V1 by V2 into equivalent weight of O2 into 1000. N1V1 is equal to N2V2. You know that. N2, how to calculate N2? N1V1 by V2 along with the equivalent weight of O2 into 1000. RQ is equal to volume of CO2 by volume of O2, weight of fish. Result, we will be seeing this result after viewing observations. Here we could see an observation: estimation of dissolved oxygen before introducing fish. Volume of water used in ml. Volume of water used in ml, 20 ml, 20 ml. Purity reading initial 30, final 31.5, 31.5, 33. So we could see volume of Na2S2O3 used 1.5, 1.5. After introducing fish, volume of water used, purity reading, volume of Na2S2O3 used. Calculations: before experiment, volume of water used 20 ml, volume of Na2S2O3 1.5 ml, normality of Na2S2O3 0.025 normal. Remember this one, 0.025 normal. Then amount of O2 present mg per ml. Amount of O2 present mg per ml N2 into V2 into equivalent weight of O2 into 1000 by V1. 0.025 into 1.5 into 8 into 1000 by 20. N2 into normality of Na2S2O3, first thing, 0.025 we just mentioned. Then V2, volume of Na2S2O3 required, that is equal to 1.5 into 8, equivalent weight of O2, 8 into 1000 by 20. 20 in the sense V1 means volume of water used. So N2 into V2 into equivalent weight of O2 into 1000 by V1 20. So after experiment, volume of water used 20, volume of Na2S2O3 used 0.5 ml, normality of Na2S2O3 0.025 normal. Same is the case with this. Amount of O2 present mg per ml 0.025 into 0.5 into 8 into 1000 by 20, 5 mg per ml. Amount of O2 used by the fish during 1 hour initial minus final value, we got the result. Okay. Then estimation of carbon dioxide. Before introducing fish, volume of water used, Burette reading initial final, volume of Na2S2O3 used. After introducing fish, we got results. Then calculations before the experiment, volume of water used is equal to 20 ml. Volume of Na2S2O3 0.1 ml. Normality of Na2S2O3 0.025 normal. So amount of CO2 present in water mg per liter V2 into N2 into equivalent of CO2 into 1000 by V1. So 0.025 into 0.1 into 44 into 1000 by 20, 0.025 normal. After the experiment, volume of water used 20 ml, volume of Na2S2O3 is equal to 0.3 ml. Normality of Na2S2O3, we get the result. Then amount of CO2 present in water 0.025 into 0.3 into 44 into 1000 by 20. We got the result. So amount of CO2 liberated by the fish during 1 hour, final minus initial value. Earlier we did initial minus final value, here reverse procedure. Okay. And after that, you got the result. Respiratory use up of organ or O2 consumed 10 mg per ml. CO2 evolved during respiration 11 mg per ml. How we got these results? We minus these results. Earlier we mentioned about the minusing. Yeah. So respiratory use up of organ or O2 consumed is equal to 10 mg per ml. CO2 evolved during respiration 11 mg per ml. Weight of the fish like this 13 gram. Respiratory quotient per gram of given sample. CO2 evolved during 1 year by O2 consumed during 1 year into weight of fish. Volume of CO2 by volume of O2 into weight of fish. Volume of CO2 by volume of O2 into weight of fish. Next, experiment estimation of total WBC count. Aim to determine the total number of WBC present in 1 mm³ of given sample of blood. So principle, blood in diluted in with a known quantity of diluting fluid in WBC. Pipette, a film of diluted blood of known thickness is spread over a counting chamber. A glass slide in which marked squares of known dimensions, the hemocytometer. The WBC was counted on with the help of microscope. The hemocytometer cover glass pipette should be clear. Then materials required: WBC diluting fluid, that is Tux fluid, hemocytometer with WBC pipette, counting chamber, lancet, cover slip, cotton and blood sample. Procedure: prick the fingertip with a lancet, wipe out first drop of blood. Suck the free flowing blood in a WBC pipette held horizontally till 0.5 mark. Wipe out the pipette. Clear. Dip vertically in the WBC diluting fluid and gently suck the fluid into mark 1. Close the top of pipette with finger and mix the contents thoroughly with a hostling motion. The capillarity of pipette is occupied by dilution fluid even after mixing, and dilution is nearly 0.5 in 100 cc in 1 m. Dilution is nearly 0.5 in 100 cc in 1 m. The cover slip is correctly placed over the bar of counting chamber. About 1/3 of the thoroughly mixed diluted blood is rejected by blocking it out of the pipette. Hold the pipette at about 1/3 of the thoroughly mixed diluted blood is rejected by blocking it out of the pipette. Hold the pipette at 45 degree angle and the slide touching the space between the groove of the side under the cover slip is just filled. In it, the air bubble is trapped under the cover slip. Remove them by using cover slip to the edges of the slab and then allow air to escape. Wait for about 3 minutes and check for even distribution of blood cells under microscope. Result: number of WBC present in mm³ of human blood. You could see the result. Next, calculation. Neubauer counting chamber. You could see like this. Calculations: number of WBC in first field. You will be writing that. Number of WBCs in second field 48. Number of WBCs in third field 61. Number of WBCs in fourth field 51. Total number of cells counted 216. Area counted 4 into 1 mm square. 4 mm square. Total area counted 4 into 1 mm square is equal to 4 mm square. Dilution factor 1 by 20. Depth factor 1 by 10. So WBC cubic mm of blood, N by dilution factor into depth factor into area counted. So N into 200 by 4. N into 200 by 4. N into 50. N was found to be 216. So N into 200 by 4. N into 200 by 4 is equal to N into 50. N into 50. How to do this? WBC cubic mm of blood is equal to N by dilution factor into depth factor into area counted. N by dilution factor into depth factor into area counted. Area counted 4, dilution factor 1 by 20, depth factor 1 by 10. Then next experiment. Estimation of RBC in human blood. We have to count the number of RBC in 1 cubic mm of blood. Principle: the blood is diluted with a known quantity of diluting fluid in RBC's pipette. A film of diluted blood of known thickness is spread over a counting chamber. A glass slip with marked spheres of known dimension. The cytometer and RBCs are counted with help of microscope. The hemocytometer cover glass and RBC pipette should be cleaned before and after the experiment. Okay, so materials required: the blood sample, hemocytometer with RBC, pipette and cover slip microscope, sterilized needle, cotton, Hems fluid, RBC diluting fluid, HAEMS. Okay, so preparation of Hems solution: NaCl plus 0.5 gram of sodium sulfate, NaCl plus 0.5 gram of sodium sulfate, 2.5 gram mercuric chloride, 0.25 gram distilled water, 100 ml. Then in heme solution, 5% NaCl is present, which is isotonic with blood. Sodium sulfate 2.5%, which prevent blood clotting, and mercuric chloride prevents bacterial growth and acts as a preservator. Once again, I am going to read this. Preparation of Hems fluid: NaCl plus 0.5 gram sodium sulfate, 2.5 gram mercuric chloride, 0.25 gram distilled water, 100 ml. In heme solution, 5% NaCl is present, which is isotonic with blood. Sodium sulfate 2.5%, which prevent blood clotting, and mercury chloride prevents bacterial growth and acts as a preservator. Procedure: the counting chamber should be cleaned with distilled water and dried. The ruled area should not be rubbed, as it is easily scratched. To the hemocytometer, the square area in the center, 1 mm square, is divided into 25 groups of small squares. That is a total of 400 small squares. 1 mm, 25. The hemocytometer, the square area in the center, 1 mm square, is divided into 25 groups of small squares. That is a total of 400 small squares, each giving 1 by 20 into 1 by 20 square mm area. Total 25 small squares, and that is a total of 400. 1 minute. The square at the center, 1 mm square, is divided into 25 groups of small squares. That is a total of 400 small square, each having 1 by 20 into 1 by 20 square mm area. The ruled area is exactly 0.1 mm below the general surface of slide. This ensures when the cover slip is placed over a film of blood filled with the chamber. The film will be thickness of 0.1 mm, so the volume of each surface will be 1 by 20 into 1 by 20 is equal to 1 by 400 or 1000. No, 400. That's a mistake. All the apparatus should be sterilized by squabbing it in the rectified spirit and heating over the flame of spirit lamp. The finger is pricked with sterilized needle so that the blood flows freely. The blood was drawn out. The apparatus should be sterilized. The finger is pricked with a sterilized needle so that blood flows freely. The blood was drawn out into a graduated portion of RBC pipette up to 0.5 by sucking it. It is then plugged to the diluting fluid and is then sucked up to not to mark. The fluid is then the pipette was mixed thoroughly. The cover slip was placed on its ruled area of hemocytometer. Descended 2 to 3 drops of mixture from pipette and a drop of large cover the ruled area on hemocytometer was introduced into it. The fluid was drawn by capillary action. The slide was left as such for 15 minutes for the corpuscles to settle down properly, avoid bubble formation, and spaces are completely filled in and examined under microscope. Then under high power objective, the counting of number of cells in 5 squares in central area, small squares of 4 corners. And high power objective, the counting of numbers of cells in 5 squares in central area of small squares of 4 corners, and on the central square of 5 diagonal squares of RBC area, each consisting of 16 squares. Total of 80 squares are done. So precautions, the counting chamber should not be over illuminated. Minimum is less. Since RBCs are the lengths of chamber are constrained, the margins of RBC standard lines can be clearly seen at minimum illumination. Before starting to count RBC, fluid is then checked whether it's uniform or not. Counted square cannot be counted again, and total counted squares should not exceed 20 while counting RBC cell will live in both the adjacent square. In such case, in which RBC come to lie on the top of the line, they are not counted. Then here we could see a calibration. Volume of the smallest square 1 by 4000 mm cube. Volume of 80 smallest squares 80 into 1 by 4000 1 by 50 mm cube. If the number of cells in 1 by 50th mm cube is x, the number of cells in 1 mm cube is equal to x into 50. The dilution of blood in 1 is 200. The number of RBCs in 1 mm cube in diluted blood is equal to x into 50 into 20. X is equal to 5 to 6. So 5 to 6 into 50 into 200. The RBC present in form of blood, we could see this. Then discussion. The average RBC present is 4.6 to 6 million cubic mm of blood of man and 4.2 to 5 million cubic mm in common. 4.2 to 5 million per cubic mm.